I'm recording.
Yes,
Okay. It does
I-
not say it's recording on my end, but
it shows it on my end.
okay. Okay, good. All right. Okay, so these are the points here that I'm going to discuss. If you have any questions during this, please feel free to just unmute and interrupt. Or yeah, I'm trying to see. I guess the chat is maybe disabled when the screen is up, the PowerPoint, because I'm not seeing that. But oh no, there is a chat. Okay, so I opened up the chat, so I'll pull that down. If you do have a question, the chat box is here, so I should see that come up. And if not, again, just let me know. So what are pollinators? Abiotic and biotic. Wind, water can pass gametes long, um, um, and of course the biotic pollinators, bees, other insects, so beetles, butterflies, hoverflies. We kind of we know what the pollinators are. Um, among animals, insects are the dominant pollinators. So here uh, it's bees, beetles, flies, butterflies, and moths. Among the insects, the bees are certainly one of the most abundant Anthophilus insects. So one of the insects that we see most on flowering plants. Okay. Um, Value of pollination. So Galai in 2009 estimated value of pollination services globally to be over 200 billion. So very important there what these insects do regarding the pollination of agricultural crops and wild plants. Over 80 crops benefit from insect pollination. 35% of total global crop production depends on pollinators. So that 35% seems low, but if we think about what crops are pollinated, those are all of our specialty crops. When we think about cotton and rice and wheat, all the cereal grains, those don't require pollination. So those are most of our agricultural production, when you think of corn and rice and whatnot. So it's the really diverse group of specialty crops that would suffer the most if we lost pollinators. So pumpkins and apples and the likes. Okay, so recently pollinator decline has been documented. That's over the past couple of decades. A few things have been highlighted with pollinator decline. One is the declines in the monarch butterfly. Last year, the Western population, I think, was reduced by about 80%. The Eastern population is, staggering. It's just the past several winters, they estimated the population just to cover a couple of hectares at most. So, back a couple of decades ago, it was not uncommon for the overwintering population down in Mexico to occupy, 15, 18 hectares. Um, these days, 1.5, 2 hectares, that's about the moving average there on the area that monarchs have been occupying in their overwintering grounds. The several entities petitioned the monarch to be listed as threatened and endangered. So that ruling was supposed to be completed in 2019. Still under review. There's a lot of data gathering going on now. There's some, special questions and activities that have been conducted. For example, the Xerxes Society, they were interested in aggregations occurring in eastern New Mexico. So, identifying areas where these aggregations are occurring that might be, during migration, but outside of the normal patterns that have been, observed during that migration process. Um, So the monarch, that, that really highlighted pollinator decline. The other big issue was honeybee decline, so colony collapse disorder. Uh, that's winter losses peaked at about 60%. So the beekeepers up in the Ohio Valley and elsewhere, they were normally, 15, 20% losses during the winter, and they were, creeping up towards 60% or so colony losses during the winter. So these colonies were stressed. Beekeepers would find piles of honeybees, or in some cases, they would find nothing. The honeybees would just abscond and leave an empty hive. So that was why they called it colony collapse. It's the colony would go from 100% to nothing. So that's a big issue, and it has been a big issue. The factors that are implicated in monarch decline, compromised nutrition. It's been estimated that over 150,000 acres, no, I'm sorry, 150 million acres of milkweed has been removed, extirpated from the Midwestern crop growing regions, because of herbicide resistant crops. So once herbicide resistant crops were created, farmers could pretty much spray bar ditches and field margins and get rid of these undesirable weeds, plants, and it really caused some problems with the milkweed. And that's the big push. That's where all their summer breeding grounds are. It's unfortunate that the biggest corn, cereal grain growing region in the country just so happens to be squarely in the region of these summer breeding grounds. So it's a tough overlap there, and but that's one of the issues that threatens the monarchs. Pesticide exposure could be another one. There's a myriad of environmental stressors that have likely impacted the monarchs. These variable weather patterns. I know a few years ago, a big winter storm moved across Tijuana, San Diego, moved from west to east, and supposedly knocked out like a million monarchs. They had just broken diapause and started flying north, and that big storm went through and knocked out a bunch of them. So a lot of different factors that are affecting monarchs, honeybees, and then a lot of things we've learned about honeybees, everything from diseases and whatnot occurring in honeybees. Those can transfer to native or to wild bees. So I know the bee lab up in Logan, they were looking at how these diseases might spill over into native bumblebee populations. A lot of tomato farmers and greenhouse farmers, they'll buy or rent bumblebees. They'll come in a little box. They're usually from this company called Copec up in Canada, and it's usually Bombus impatiens, which is a little tiny bee. Well, that's introduced into the environment, and if they have any problems, then those can be spread to the native populations.
I have a quick
Just a
question.
little bit more on pollinator decline and the implications. There's been some research going on that has been looking at, okay, what wild bees are visiting crops, and what portion of the pollinator community is actively contributing to those pollination services in crops. And it's really been found that, at least in this study here in 2015, that there's a few very common, abundant, dominant species that, numerically dominant species that are visiting crops, and therefore the conservation of pollinator biodiversity might not be in line with the conservation of pollination services for crops. And I really think those are kind of two different things and need to be addressed in two different ways. You know, because we do have a lot of specialist bees and a pretty vast bee biodiversity, you know, but how many of those are actively visiting these crops? This paper suggests it's likely a few, which, you know, you can look at that a couple of ways there. You know, one is that these native bees are providing free pollination services, you know, so that's good. Maybe it's a halictus or a common sweat bee. On the other hand, if a large majority of the bee biodiversity isn't visiting the crop, then maybe that affords them a level of protection from agrochemical exposure and things like that. So, you know, we're in a good place to study these things, you know, because of the landscape matrix, because of the land use practices. You know, we can go out to little remnant grasslands and collect bees and study them and see if anything in the agricultural realm is affecting them. We actually published a paper last year. We collected about 300 native bees from little grassland remnants around crops, and we measured agrochemicals. So we were interested in what agrochemicals, especially the neonicotinoid compounds. We were interested in what were those on these native bees, and we found 20 out of almost 300 bees. 20 individuals had residual neonicotinoids on their bodies.
Hey, Scott,
Yes.
we have a member held up in the waiting room. Could
Oh,
you let her in?
I sure can.
And I just wanted to draw your attention to some questions coming in on the chat.
Oh, good. Okay. Well, we'll transition here in a couple of slides. So I'll go ahead and take those questions. Is colony collapse disorder affecting wild bees too, or mostly commercial beekeeping? So, you know, by nature, colony collapse disorder affects honeybees, and a honeybee is a social bee. So there's different degrees of sociality in insects, all the way from entirely eusocial bees to solitary bees. So the colony collapse disorder only affects the honeybees, as far as we know at this point, because the honeybees build up these big colonies, and therefore they are susceptible to these issues, to these diseases, because they have the numbers, they have the contact within the hive, they have all the workers communicating through chemicals and touch, and it's a very very abundant nest. So no, it's not affecting wild bees as far as we know right now. Although I think with bumblebees, there are some pests and things that can be transferred, you know, with these species sharing the same plant, basically sharing the same flower. So if a diseased bee flies to a flower, then it makes sense that there's potential there for that to be transferred. Our commercial bee, another question is, are commercial bee suppliers careless about testing for disease? Um, um, so I think that's in regards to the bumblebees I mentioned. I don't know, honestly. I don't know what their protocols are for testing. I do know that they rear out a lot of bees for commercial pollination. I don't know if they're careless or not. That's a good question, but I would be surprised if they didn't have protocols in place to do some sort of random testing before those bees left their facility. Okay, the next slide shows native bee, an example of a soil nesting bee. So about 70% of our bees, we have about 4,000 wild bee species in the US, about 900 species in Texas, and over 75% of these nest in the ground. So there's all sorts of um, conservation implications there and ecological implications regarding the quality of nests, the persistence of nests, soil disturbance. So, while disturbance can, I think, benefit bees in some ways by, exposing bare soil and um, um, lost my train of thought there. So the structure of these nests. This is an example of a solitary nest, solitary bee. So bees can, like I say, they can be eusocial, so they build colonies. Like, we're hearing a lot about the Asian giant hornets, and we have the European giant hornets already in the States. Those are social bees, or social wasps. Uh, Honeybees are social. Bumblebees are social, although bumblebees, they're not a perennial colony. One female will start the colony each year, similar to wasps. Whereas honeybees, they'll just hunker down and warm up the queen all winter, and then they'll have that um, core of workforce of workers um, um, to gather pollen and whatnot. A bumblebee has to do that every year. Our solitary bees, they can range from strictly solitary, so one queen is entering this tunnel, laying an egg and provisioning pollen. Some of our halictids and some of the other bees, they can be communal or they can aggregate. So multiple females, they all lay eggs, but multiple females can share the same nest. So that's not social, but that's a communal. They're using that area to nest, and a lot of our sunflower bees and other bees will do the same thing. So when you see a soil nest and you see a bee flying in and out, likely there's an aggregation of nests there. Um, One question I have regarding the soil nest is that what we study the agrochemicals on bees. Well, in places where these bees are nesting, are agrochemicals, are they being translocated from the source into the soil here? And if you think about it, if bees can be contaminated on a flower or any other vegetation, then it makes sense that those chemicals might end up in the soil in some concentration. So that's another research question that we have been looking at. So out of the 4,000 bees we have in the U.S., about 35 to 50% will specialize on only one family of plants. So that suggests, and that's especially prevalent in the Southwest, where we have, a really high bee biodiversity. We have some of these desert type plants that can provide these resources. Prickly pear cactus, desert globe mallow has bees that are specialists. Coreopsis, Namahispidum or sandbell has a group of bees that will only forage on that plant. So, out across the bee biodiversity, a third or more of the bees are looking for one type of plant, looking for that genus of plant to forage. Okay, and if you, I'm sure you all have witnessed opuntia and leaf cutters and honeybees. It's probably one of the coolest, most interesting sights I see during the year is when the prickly pear is really putting off the pollen, and the honeybees and leaf cutters and all sorts of bees are just kind of diving in and out of the flowers. It's a neat sight to see. Here on the high plains, we really, before we started the research and before a few other studies, there's been a couple of studies here in the last few years that have complemented our work. And among these studies, we've really begun to better understand the native bee biodiversity in the region. And it's actually, it's much better than we thought. We go out and we collect in grasslands that look just like this, and we'll put our bee bowls out, and it's kind of amazing to put a bee bowl out and not have any flowers around. Come back the next day and there are 30 or 40, bees in the cup. We saw that down in South Texas too. We saw it in cornfields where we were putting these bee bowls out in cornfields and getting a pretty abundant sample of bees. And we were kind of wondering what was going on there, whether they're using the soil or whether they might be using some of the resources from corn. Okay, so basically we just didn't know a whole lot about bees before the last few years. I had a student who's defending actually on the 16th of this month who studied native bee biodiversity and worked on a project that we had funded from the USDA to go around to different farms and sample bees. We had a goal at each of these farms uh, to conduct active restoration. We have a little 40 horsepower tractor and a seed drill, and we were going around to all these farms and drilling seed into the soil. We were doing it a lot in unirrigated locations. So once, that lends itself to some problems, because rain is so variable. So in some places we put seed down and it, didn't do anything. In other places, um, a farmer might have had, some irrigation access or something like that, where, all it takes is a little bit of water and we see good results. But that's, that really doesn't happen in the wild areas. I have a question here. What are bee bowls? Thank you for that question. That's a good question. The bee bowls are little, they're very simple. They're 3.5 ounce little cups, like little condiment cups. We get them from a place in Maryland um, called New Horizons. If you're interested, it's a really good company. I learned about it a while back. Uh, they have handicapped and disadvantaged people that paint them fluorescent colors. So every year I order, you know, quite a few bee bowls from them. A bee bowl is 3.5 ounces. It's painted either fluorescent yellow, fluorescent blue, or white. So they use a fluorescent paint, which is attractive to the bees. We fill the bee bowls up with a soapy water solution. The soap in the water breaks the surface tension, so a bee doesn't land on the water and then fly out. So those are the bee bowls. And bee bowls are biased towards a certain group of bees. So if you put a bee bowl out, if you put a little yellow bowl out, it's likely going to collect a lot of um, halictids, a lot of sweat bees. Christopher just posted the link. Yes, the link to New Horizons. Okay, so it's New Horizons. If you ever want to get bee bowls, I encourage you to order from them. They're great. They usually have them ready to go, and if not, they'll get on it and they'll crank out 1,000 of them in no time. Okay. Um, okay, so let's move on. This is one of the areas that we conducted this active restoration. This is just, we used a seed drill. We drilled the seed. This one on the left here, we had 5 species of native grasses in the mix, and then a 30 species mix of forbs. So, Gaillardia kind of showed out here this first year. It sort of mass emerged, and over time the grasses kind of filled in and, kind of made for a nice little habitat that we were able to work in. So this is how we sampled across the farms. We conducted standard transects, which was a 60-meter transect, 2 meters wide. We sampled within that. We also set out bee bowls, and then we conducted a random walk transect, and that was so we could locate flowers. It's also called hunt sampling. So if you conduct hunt sampling over a 30-minute time period, that means you go randomly walk and you search for flowers. So, these standard transects, we set up a 60-meter transect. The odds of getting, flowers that might be missed within that linear transect is pretty high. So that's why we did the random walk transects. And we sampled in different seasons. So this is just kind of a synopsis of what we found here. And this was a little early on, so some of these numbers have changed a little bit. So about 17,000 bees. This genera has risen above that 49, but 5 families, from 64 to 615 bees per site, an average of 10 to 21 genera per site. Lasioglossum, a little sweat bee, was by far the most dominant bee. This is a species complex. Very few bee taxonomists in the country really can identify these Lasioglossum past this genus species complex level here. There's, like with a lot of bees, there needs to be some revisions and clarification of the taxon. But sweat bees by far dominate the high plains. So this shows the yellow bar here. Those are the halictids. Those are the sweat bees. By far the most speciose family of native bees in the area. So andrenids, apidae, these are the honeybees, coletids. We have a couple of genera of those. Halictidae, then the megachielidae are the leaf cutters, the leaf cutter bees that I'll discuss a little bit more. Okay, so the next 3 slides are just a synopsis of what we found. And this, my student wrote a checklist. His last chapter of his dissertation was a checklist of all the genera and species across this 14 county region. And this was, it wasn't a formal meta-analysis, but he included information from 2 other studies that had been conducted. One at Muleshoe Wildlife Management Area that was conducted over a couple of years, and then another one conducted by a student, Angie Bigosh from Oklahoma State, that was looking at differences in bee communities from playa basins to uplands. So we used our sampling, the Muleshoe study, and then this Bigosh study to come up with these summaries, which are probably the most accurate account of what we know about native bees in the southern high plains. So 286 bee species in a 14 county region. This is likely an underestimate. For example, John Mack over in Travis County, he's documented 299 species at the Brackenridge Field Lab there in Austin. So that's just an example. 286 species across 14 counties. In that one county, they're up to 299. So we have an idea that we're undercounting right now, but then we have a lot of questions too. So those species that were collected previously and we haven't collected now, is that because we're not detecting them, or is that because they have been extirpated from the area? Okay. All righty. So 3 species account for 30 to 40% of the species collected to date. That's Agapostemon angelicus, a little super bright, fairly large green sweat bee, Melisodes coreopsis, and then Melisodes communis. Those are the 3 numerically dominant species in the region. This slide gives an example of or gives a listing of the families. So these are the main families that we have, or they are the families we have here in the High Plains. So Apidae, Andrenids, Megachilidae, Halictidae, Coletidae. The Apidae are the most diverse, whereas the Halictidae are typically the most abundant. Okay. Although with, like feral honeybee colonies, we don't exactly know, it's hard to count honeybees. They can certainly be abundant, and we really don't have a good handle on how many feral colonies might be roaming around out there. But certainly they are unaccounted for. So anyway, the 5 families there, 158 species were found in addition to 128 species previously reported for the region. So this was what we knew. 128 species is what was reported. So we added, we and then the 2 other recent studies have added 158 species. Wow. Only 10 species reported for all 14 counties. So what does that say? Who knows? That's the ubiquity of those common species. That's behind it, because some of those sweat bees are fairly dominant, but still you would think that there would be more than 10 species occurring across the entire region there. 92 specimens were reported from only 1 county, 51 with only a single voucher specimen. So, what does that say? Does that say, well, are those specialists? Does it say they're hard to detect, or indeed their occupancy is really low? They're not there. Detection can be a problem, especially with the specialist, because if you don't find the plant, you don't find the bee. So, one reason people like Dr. John Neff are so good with this stuff is because they know the plants as well. So they can locate XYZ plants and go, stake out at the plant and know the bee is going to come by. I'm certainly not that good in terms of the plant info. So we're learning really a lot as we go, but it's a little tougher when you don't know, exactly all the plant biodiversity. Okay. 3 species not collected over an 80-year span, a Nomata species, and then a Melisodes, and then an Andrena. What does that mean? Again, we don't know. If we were, to find out the resources that these bees use and investigate that a little further, maybe that would show us something a little more informative there. But it is telling that these 3 studies all came up empty for these taxa here, these 3 species. And these are the studies here. Begosh study, I don't know if any of that has been published yet, but that was the dissertation out of OSU. The Auerbach paper was the Muleshoe Wildlife Management Area assessment, and then DISCWA, that was the studies we conducted recently here from the USDA project. This figure shows, the blue bars show the historic records. The orange bars show what we've added per county in species records. Okay, so that's, this figure here says a lot about what we're doing, and that's, we're trying to get some background data, some meaningful data here, so we can, conduct further monitoring, so we can start looking at, specific crops or specific plants. What are the bees that are visiting the crops most? This gives us a good background to build from. So we'll continue to add to these records. The next study I want to show you here is a cavity nesting bee study. This was conducted by my master student, Caleb Tomlinson, who should be finishing up this semester. So we were interested in really, kind of taking the piece of the pie of the cavity nesters out of the bee community and focusing on that fauna, and really digging our heels into the taxonomy of this group. So what species do we have? And then secondly, when we put out these cavity nesting bee traps, what's the fauna that shows up? And we were, we knew we wouldn't get only bees, but we were kind of surprised at what we found. So this is what one of these cavity nests would look like. Okay, so this is a leaf cutter bee. This looks like a megachile, so a native leaf cutter. So this female, one female will use one hole, and this female will go in and provision pollen. Uh, we'll make little brood chambers. We'll mass provision pollen. The female will lay egg on the pollen mass, and then that egg will hatch. The first instar larva will start eating that pollen mass. And then the later instar larva will pupate in this cell. So they don't leave this cell. So it's kind of a low risk, besides, the potential constituents that come back in the food, which these things are chock full of agrochemicals. Besides that, this is a pretty low risk, the foods mass provision, the eggs laid, the larva eats the egg, and then the new adult will emerge out. Okay. Usually these brood chambers are lined by plant material, in some cases mud and other materials. But usually these leaf cutters will use pieces of leaf material. I used to have a lot of 4 o'clocks in my home garden. Uh, Before we moved, I act like 4 o'clocks go away, but they don't. They just sprout back up. They love 4 o'clock leaves. They love trumpet vine. That's what I've seen them clip out the most. Roses, things like that. So, not a super thick leaf, but something that's pretty easy to work with. Okay. Um, Okay, so a little bit more about the leaf cutters. They're a little different in that they don't carry, some non-leaf cutter bees will carry pollen internally, or they'll carry it on their legs, or pollen baskets like in the honeybees. These leaf cutter bees, they'll usually have a bunch of hairs on the ventral surface of the abdomen, where they will brush pollen back and then carry the pollen right here on the scopa. That is the pollen basket, and therefore they can usually carry a pretty big load of pollen. And why are these bees important? Here we have the foraging distance of bees. So we know that a bumblebee, if it's good and hungry and looking for resources, it can travel up to 8 or so miles. A honeybee can also travel up to 8 or so miles. If it's really looking for food, although a honeybee is generally, it's going to stay within a couple of miles. These mason bees and leafcutter bees, they're more localized. They only go a few hundred meters at most. Okay, so the leafcutters, it's a pretty drastically different foraging distance. What does that matter? Well, it matters in terms of the local and landscape level habitat. So what plants are around that are benefiting from these mason and leafcutter bees, that's going to be in a more restricted zone. And it also might have some implications in crop pollination. So I was contacted recently about doing some research in sesame. So this particular group is looking to increase the hybridization of sesame. So, they're wanting to increase the outcross pollination up to 30 to 50 or more percent. And they're wondering what sort of native bees might lead to that sort of outcross pollination. So, we have to take into account a lot of different things there. Most importantly, what bees are visiting the sesame, but also, a sesame has both a male and female part within the flower. So, there's some size issues there maybe, and uh, the action or just the foraging behavior of a honeybee is much different than a native bee. A honeybee, there might be 3 or 4 native bees on a flower, and if a honeybee wants in there, it just bombs the flower and just kind of throws them all out of the way. So There's differences in foraging and differences in traits and how they forage on that flower. So those have to be taken into account when looking at pollination. So the trap nest, I'll show you on the next slide. They were placed at 21 locations across the high plains. We had many different sites, but everyone knows pretty much how heterogeneous the landscape is around here. So about any place we clip out here is going to have a portion of crop, a portion of maybe a pivot corner, maybe a bar ditch. We've done some of the preliminary analyses and found that on average, any place you clip out, maybe except if you're in eastern Lubbock County where there's just kind of wall-to-wall crops, 20 to 30% non-crops, what we call grassland or open habitat is kind of the average. So, which is good because studies out in California have shown that if you keep 30% of your farm in wild land, then that can provide the pollination services for, I think they were looking at a watermelon crop. So, and they suggested that, okay, farm away, but if you leave 30% in wild land, then you might have the habitat available there to support pollinators to provide pollination services in that crop. So these are the nests we put out here. We got these, we received these, what we called layered nest from Crown Bees. So it's a company. They have a nice little, uh, uh, drill press, and they can make all these nice little even 5/8 holes here. Okay, so that's, if somebody's interested in leaf cutters, or if an orchard wants to increase the habitat available for nesting species, they might put out some of these nests, or you might order Megachile rotunda, blue orchard bee, something like that, release into an orchard, and then they'll come back and they'll nest in these habitats, in these cavity nests. If they're not nesting here, they're building their own nest in trees or wherever they can find. So the other photo here shows what we call the drilled nests. Okay, so these are our homemade nests. We had 4x4 posts, and we had drill bits, and we drilled these nests in different diameters. So 5/16, 7/32, 5/32, and then 1/8. Okay, we just simply wanted to vary the nest diameter holes so we could see what sort of species were occupying those. Okay. Okay, this is what we found. So we found pretty much an even, more or less even percent colonization across the different holes, which was a little bit unexpected. We thought these 5/32 would be a little more, or the 5/16 would be a little more colonized. But we found that the smaller sized holes, especially the 5/32, had a higher colonization, which surprised us. So we reared out 729 individual insects. So my student collected these nests, brought them back, created a little chamber to isolate this size hole, and then basically just let everything emerge. And then as things emerged, this student collected everything, and then ID'd everything. Okay. So this is what, this is what we found. And this is just kind of an opposite pattern here. So the drilled nest, the homemade nests, had 76.8% bees and then 23% non-bees, which is about typical. However, the layered nest had over 77% non-bees and then about 22% bees. What does this say? This says, well, if you're wanting bees, a cheaper route, a less expensive route, and a better route is to drill these 5/32 holes or smaller, around that size, and provide that for these bees. These layered nests, we're not sure why they attracted so many different non-bees and wasps and spiders. but they did. We did build a box. If you go back and you look at this nest, what we had to do, we had to build this box that contains the layers. So these are individual pieces of board. You know, one row of circles splits two pieces. And then we put a little, you know, we made these, so we had to put a little shim in there to make sure they were good and tight. So we think just the addition of this box, which you have to have some sort of box to put these things in, but we think just the addition of this box might have caused some of that non-bee colonization. In any case, so these were our drilled nest species. So Ashmediella bucconus, a little megachilid, that was the most dominant bee. And this one mostly occupied that 532nd hole. And it was the most dominant bee. So it wasn't what we thought it would be. It was this little tiny leaf cutter. And it was really abundant, 354 individuals, followed by, some of the megachile, more common ones here. And then a couple of eudonerous bees, or wasps, I'm sorry. And then we have some flies. We have a lot of flies. We had a beetle in there, this little a beetle. So that's what we found. We're basically wanting these, but getting these, we also get a lot of parasitic and non-bee species. This is also shown in the literature. We knew that we would find non-bee species. We didn't know exactly the diversity we would find, but this is what, if you put a drilled piece of wood out, these are the types of cavity nesters that are going to be finding those nests. So, it also sort of begs the question, what's the role of coarse woody debris here on this landscape in these grasslands? You know, just about every week we're driving out and we see old house places that are abandoned, that they might be used for chemical mixing yards for farmers or whatever, but I think we can all probably think of a few house places that have a lot of old trees that are down, and that sort of habitat might be really undervalued in this region, especially because it's predominantly a grassland. But that's what these species are looking for. They're looking for wood and cavities to nest in, to either parasitize or to raise brood. So it's a pretty favorable habitat, it seems, by this community here. So these are the layered community of the layered nest. So this is the commercial nest. So the dominant species within this nest was not even a bee at all. Actually, the top two. So Eudonerus and then this Pollinia fly here. So these are, the top two species weren't even bees in those nests. What we would expect to see would be Polycharis or these other larger megachiles. This graph just shows some patterns according to the hole sizes. So on the x-axis here, if there's an L, so the layered nest, this is the commercial nest here, the layered nest, okay? The first bar is the commercial nest. The next 4 bars are the drilled, okay? So these are the drilled sizes. So we see that this 532nd hole had more individuals for the bee species. The non-bee species, the layered nests, like we just saw, had more non-bee species, okay? Number of individuals of the parasitic species, so all of the larger sized holes had a high proportion of parasitic species. Nest size count, so these are just total number of individuals per nest hole. And again, 532nd is a little higher because of Ashmediella. And then the number of species, these are the nest size counts here based on nest size, and these are the species. So the total richness of the total number of species coming out of each hole. Okay. The layered nest had more diversity because of that high proportion of non-bees and parasitic species. So these next three slides just go through the sizes. So this is the 5/16, okay? That's the layered 5/16 hole and the drilled 5/16 hole combined. So you can see here, wasps fly, there's a couple of bees, but a lot of colonization by non-bee taxa. 7:30 seconds. So here's where Ashmediella starts becoming a little more abundant. So 83 here. So it appears that this species is both highly abundant, dominates some of these cavity nesting communities, and prefers these smaller hole sizes. So here's 155 total individuals of Ashmediella bucconus was pulled from those 5/32 holes. Okay. And then the 1/8, a little less still, Ashmediella is still used in that size. And then a few more here. Okay. So the next thing I want to discuss is a little patch of land that I've been working with a couple of colleagues on documenting the biodiversity of this little wild patch. So if you're familiar with 4th Street and Quaker, this is the intersection here. There's Sonic and Roses there. So this, I think, was the old Tech Rodeo Arena. We have a few publications, or actually a few photos from the 1940s and 1950s. And this place really hasn't changed. Back in that time frame, we think they were doing some agricultural studies looking at cow manure and using dug playa basins as sources of water to grow biomass. We're kind of piecing it together about what was going on this land back then. But in any case, it's a very topographically diverse area, and that's kind of why I like it. There's a lot of little different types of habitats out here. There's 3 dug playas, and then there's 1 natural playa, what I call a natural playa, but it's a playa basin that was sort of robbed of its water drainage when 4th Street was built. And so all the water's captured over here on this side. And this is a kind of a perennial system. And then over here is just a kind of a playa bed, but there's a lot of playa vegetation, smartweed and sunflowers and whatnot. So anyway, I've been kicking around out here on this land for several years now. I've actually filled out an application to reserve this space just for student education and research, and sort of just to get students out here. We're only, we're a half a mile from campus here. So to get students here, it's really easy to do. And usually, there's enough out here that it can keep us busy really for a semester. We've had several outdoor labs here this fall, and we've not gone anywhere else. So it's a really good outdoor lab. We're learning a lot about the place. We experienced the big rabbit die-off this spring. I've never seen so many dead rabbits ever, but everywhere we looked out there, this, starting about January, February, the big rabbit hemorrhagic disease that swept through some of the western states, it hit us this year. And, we had rabbits all over the farm, and it went from hundreds to 0 in just a matter of, weeks. So that was an interesting time. So that was this spring, and we've been continuing to do some work out there. This is what it looks like. This is that old playa basin. This is all smartweed. There's a very interesting little drainage that kind of backflows off of this, past this levee, and then there's a dug playa on the other side of this levee. And in wet years, it stays pretty wet, and there's mallards and everything else on it. this ply here will hold some water, but usually just on this end, and it usually will be captured by this Doug ply pretty quick. But so, you know, kind of an artificial system, but certainly one that has some diversity um, in at least in the urban area. So this is an example of, this is one of the Doug plies. Um, you know, there's some weedy pressure and whatnot, but it's pretty wild. 4th Street runs over here. Here's a photo of this is that perennial ply across the levee. So this was 2 years ago. So this year, you know, it was a very dry year. This thing dried up last winter and didn't fill back up. Um, you know, who knows what'll happen next year, but in wet years, you know, I can take my class here and we can collect diving beetles and do all sorts of stuff there. So it makes a good little wet season habitat. So here's a photo of my class, entomology class. We were just sweeping the vegetation here at the margins. Um, There's a big hornworm, a big Saturniid um, caterpillar. So the students, you know, students see things out there that otherwise, they might not see. It's a very convenient location to take them to, you know, they can get there quickly, and there's certainly a lot to show them out there. So it's been a treat. There's a little diving beetle larva we pulled out of this the sample, that aquatic sample. So, you know, some of these diving beetle larvae can get really big and eat, you know, there's photos of them eating, you know, adult dragonflies. So a little aquatic predator. This was another uh, interesting creature we found here. This is Belostoma. This is in that giant water bug family, Belostomatidae. So the females will lay eggs on the back of the male for protection, and then the male will carry those eggs around to protect them. Um, so anyway, you know, students, otherwise they wouldn't see things like this. So it's a little neat little piece of land. One of the best things about the property is that a couple of colleagues, Catherine Galley and Carl Sequest, have been very frequently visiting the site and documenting what's occurring at the site, which has been a huge source of information, you know, to figure out what's there. We have some ideas moving on down the road about looking at citizen science versus more quantitative sampling. So, you know, what do we gain from citizen science from an ecology and conservation standpoint? In some ways, I think with directed training, I think citizen science could even be more effective than, you know, empirical scientific studies, especially when we're looking at specialized taxa, simply because, you know, we can instruct and we can show plants and show bees, and you know, that might have a low detection probability. They might be hard to detect with normal type of quantitative sampling, but with, you know, focused hunt sampling, you know, that uh, can be a better way to sample these, at least for defining, you know, their status and distribution. Uh, so Catherine has started up a nice project here on iNaturalist. So I'm not sure how many, well, here we go, observations, you know, over 1,600 observations. Um, what we plan to, or what I plan to do here immediately, we have a patch out on this land that's been shredded, and I'm going to drill seed this fall and try to increase the floral diversity there, and then continue to take photographic observations and continue to monitor this area and see if we can detect any change through citizen science. Um, so that's one immediate question we have there once we begin some of the active restoration at this site. So this is kind of this little, here's this uh, greater Lubbock area here and some observations, but we found some aggregations, some bee nests out there. So Diadasia, Savastra, Those are two bee genera that are actively nesting out in these locations. Um, the first nest that Carl and Catherine found was Diadasia, and it was in an area that was just on the north side of the new electrical substation they built. There was a bunch of sort of unpacked and unconsolidated soil, and the bees nested in that, probably because it was a little bit unconsolidated and easy to tunnel in. But then we had a big rain, and the rain washed some sediment over this big nesting aggregation, and we don't know if that affected them or not, but it certainly could have caused some problems with that little population. But so far, we've identified 3 different areas where these active nests are being built. I tried to upload a video of one of them and couldn't get it uploaded, but it was a sight. This summer, um, Catherine located a aggregation of these Savastra bees, and there was also 2 species of um, parasitic bees, and they were all going at it at the same time. The parasitic ones were attacking the sunflower bees, and there was some mating going on with the sunflower bees. So it was just kind of a crazy thing to see, but very neat to see, and you know, uh, pretty cool that it's happening here in this little urban piece of wild land that's just sort of neglected. Okay, let me go back out. Okay, okay. Um, okay, so I'm gonna finish just on talking about one little specialized bee that we've been studying. We actually did start a little project on iNaturalist. I think it's called NEMA. Uh, um, so we're locating these patches of NEMA, uh, and beyond that, we're documenting the species that occur on this plant. So NEMA is specialized plant. There's a group of bees that will only forage on NEMA. This bee here that's in the photos is one of them. So this is Sphacotisoma prati. It's a little Rhophatini subfamily bee. This is the male here, the darker colored, longer antenna. Um, the female here with her head down in the corolla. The males act, they just harass and harass to no end when these bees are foraging. They'll fly by at full speed and contact the bee and keep going, or try to mate, which just happens in a very short amount of time. But if you find this plant in midsummer, it starts blooming about April. The bees come out shortly after, and if you're lucky enough to see this plant, usually you'll see this species, and you'll probably see 2 other species that are specialists on this plant. The flower here is about 7 millimeters across, so that gives you an indication of the size of this bee. They're very tiny, but they make up for it with a lot of character. So what's interesting with this plant is that the distribution is in the southwestern U.S. 2 species overlap somewhat, Nema hispidum and Nema stevensi. Both of these occur. This paper here states that the only place that these 2 have been found to occur together is up here on the Red River. Gives a location, but throughout the rest of the range, it's a fairly distinct separation here of stevensi and hispidum. Although, this is a 1984 publication, it would be interesting to go and resurvey for this plant to kind of see what an updated distribution might be. But in any case, this is a specialist bee, specialist plant system. Um, I've talked to people that know this plant, and over in New Mexico and Arizona, it's called purple mat because it'll grow in big swaths and sort of make the ground purple. And certainly the bees are probably there. When we get over here to the high plains, because of all the agricultural production, who knows what has happened to these populations of both the plant and the bee. Luckily, the plant itself is probably the toughest plant I've ever uh, uh, been involved with. Um, it likes disturbance. So if the city or the highway department goes in and creates a new sidewalk or disturbs an area of the roadside, this silly plant will grow up there. It produces thousands of super tiny seeds, and it does well in disturbance. The one of the best populations that I've seen of this plant, besides Lubbock Lake Landmark, was actually in the where the bison aggregate at Caprock Canyon State Park. Um, I was kind of blown away when uh, I took my family over there and we, pulled up there to where the bison kind of aggregate before you drop into the canyon area. And all over that little, it looks kind of like a corral. There's no vegetation, but where there's a little bit of protection with a fence or a tree or something, there's Nema growing, so it likes, it doesn't like competition. It likes to have open area that is disturbed. And odds are that these little tiny sinks are all over the seed bank. So when you do disturb, it can come up. Um, I I tilled in that restored grassland I showed you earlier a couple of years ago just for kicks. I tilled 6 patches into the grass just to see if Nema would come up. I didn't know of any seed there. I didn't know what the seed bank was. This field was an old fallow field, and I did I did 6 strips, 5 foot wide each, and about 50 feet long. And Nema came up in 3 of those patches. So, this plant needs disturbance. It wants disturbance. It thrives in it, and it supports native bees. So, the question of how has landscape disturbance shaped, the populations of that plant is an interesting question. So here's this. This is directly below one of these Nema plants. Here's a flower, and these bees, Sphacotisoma prati, forages on this plant and then nest directly below the plant. I just happened to see when I uh, enlarged this photo, this little spider here. And what's this little spider doing? I don't know. It could be ambushing, newly emerged bees. Who knows? Uh, but these are the size holes here. You can see that the diameter of this hole is just about the same diameter of this corolla of this Nema flower. But these bees nest just right below the plant, which you don't really see, in any bees, except these specialists. So, these bees forage and they just go directly below the plant and nest if the soil is suitable. So it's a very, intimate, very tightly linked system here, and that we're just really starting to learn about. These are what some of the nest would look like here. There's a little nest there, little nest there. Again, these little piles here are nests. This was an interesting photo because this Nema plant was completely dead, and this Nema plant was, as you see it, uh, doing okay here. There are each one of these little scree piles, tamuli, are that's a nest. So can nesting affect the plant? Who knows? That's a question that we'll need to look at. These are females nesting down into the corolla there. You can tell the Sphacotisoma prati, it's a black bee with a red metasoma, or red abdomen here, and these little white fringes of hair here. You can see these little white fringes of hair. That kind of is what gives it away within this subfamily. And then here, actually, if you look at this plant, there's a bee there, there's a bee there, and there's, I think, a bee there. So, that's just showing you that usually you see these plants in the summer, these bees are all over the plant. So how can citizen science assist, for example, this species? We can learn a lot just by charging citizen scientists to, find this plant and this bee. We asked last summer if bee bowls would pick up Sphacotisoma prati. If we're monitoring for this species, can trapping pick up this bee? So we went to Home Depot and we matched the color purple the best we could. And we got our own bee bowls and we painted them purple. And we put them right next to NEMA. Went back a few hours later and the Sphacotisoma prati was in the purple cups next to the NEMA. So they are attracted to that purple color and they are going into the bee bowl. So we can use bee bowls to detect the presence of this species if we need to. Like I say, usually the detection and the occupancy of this bee is usually high. You see the plant, odds are if you go look at the plant, you're going to see the bee. But, for monitoring, if we put out purple bee bowls, we do know those are effective too. So, so we can use those for monitoring if needed. Moving forward with my research, I'm very interested in citizen science and how these empirical studies relate to citizen science, what we might can achieve from citizen science that we don't get from these big biodiversity studies. Which the big study at Muleshoe that they did a few years ago, that's the study. That's kind of the study. The Fish and Wildlife Service conducted that. They used blue vein traps, which are supposed to collect the most number of bees and the most diversity. And this study was over 2 years. So I eagerly grabbed their data set. It was available at one point, and I just wanted to take a look at the species list. And Sphacotisoma prati was not anywhere on that list. It was not in the database. So, that tells me that, these bigger type studies, likely are missing some of this very important biodiversity that, that we might, it's going to be tough to document just with traditional type studies. And I think citizen science is a way to advance, what we know about the distributions of these species. Okay, I'm going to end right there, and I neglected the chat, but I'm going to open up the chat right now. So Lena had a question. How did you determine bee species? Physical characteristics? For Sphacotisoma prati and all the other bees, we used physical characteristics. So just character traits. We also used experts, Dr. John Neff in Austin, and Dr. Karen Wright, who's a bee taxonomist with Texas A&M. They are way better bee taxonomists than I am. My training is in aquatic insects or a lot of my training is and research is in aquatic insects and diving beetles and things like that. Bee taxonomy is fairly new to me, so I consider myself a novice, and I'm certainly learning a lot from others. My previous student, Samuel, he knows more than I do about native bees. He took we sent him out to the native bee course out in Arizona a few years ago, and so he got up to speed pretty good on that. But we mostly, so far, we haven't done any genetics analyses, but once we start looking at population level studies, I think it would be very interesting to start looking at population sizes versus habitat characteristics and habitat fragmentation. So, it's likely that NEMA hispidum is very fragmented as far as its historical distribution, and really even its ability to, to disperse and to, disperse during maybe soil disturbance events or whatnot. it's soil disturbance certainly happens in ag lands, but it's a very unnatural system. So uh, the dynamics of those populations are interested, and the dynamics of the native bees are interested, especially like their effective population size and other population conditions that can be determined genetically. I think that's when we can start looking at these populations and saying, okay, X amount of landscape fragmentation affected this population in this way. And then, that feeds back into knowing about how we can restore lands and manage lands so we can improve that biodiversity. Okay, information on rabbit hemorrhagic disease. If anyone is curious, Christopher posted a link there. So yeah, we were right in the middle of that. What was going on there, and I'm sure if many of you were out, you probably saw dead rabbits too. But it was well, it wasn't funny. It was serious. But just as the time COVID hit, you know, we're walking around out on these lands, and literally everywhere you turn, there's a rabbit carcass. So it was a kind of a weird time, worrying about COVID, and then everywhere you look, there's another dead rabbit. And then what really got our attention was a big red-tailed hawk that was dead about 3 feet away from one of the dead rabbits. And that, when we saw that sort of changed things. We don't know the cause of the death of that hawk, but maybe it was related. Okay, Cheryl said, are there more slides? That might have been when I went to full view and it clipped those out. Thank you, Sienna. Have you set any bee bowls in the old pecan orchards like at Brownfield? I have not. We just got a new faculty in NRM, and they're interested in pecan orchards and improving the cover crops or using cover crops in pecans. So there has been some discussion started about sampling in pecan orchards. So Dr. Caitlin Cooper in NRM might be heading up some of that work. Okay, well, everybody is certainly welcome. I feel like I kind of rushed through that. If you have any questions, or have any questions now or down the road, if you want to email me, I know all of you are interested in nature and maybe citizen science, and if you keep in touch and want to know more about what we're doing, please do. Not that I can tell you exactly what we're doing X, Y, Z, but there's a body of research. If you do a little lit review on citizen science and insects, there's a growing body of literature that is really kind of steering the the way in which citizen science can really assist conservation. And I think it's underrated or under appreciated at the moment. So with a bee like Sphacotisoma prati, that's I think one species that could certainly benefit from citizen science. So we're in the process of writing a little short paper, maybe a note on this scenario: name a hispidum and Sphacotisoma prati. What's the conditions there, and what makes it a good species to engage citizen science? I think Susan is raising her hand and might have a question.
Yeah, I do.
Okay.
I work in the nursery and landscape industry. What I'm wondering is, like, when you use a systemic insecticide on a plant, is that going to affect a bee?
It can, and it can do that in two ways. So you probably know that all the cotton is treated, is seed treated, right?
Yeah.
And those are all systemic. systemic insecticides and fungicides, they want that seed to not rot in the ground or not get eaten. So that seed takes up those systemic neonicotinoids. Whether those neonicotinoids are expressed in pollen and nectar and in the, constituents within the bee resources, that's an area of debate. There's been studies that show there is no translocation of those chemicals up into the bee resources, the nectar and the pollen. And then there have been some studies that have shown there can be some contamination. There was a big study in Arkansas done, I think back in 2015 and 2016, and they did not find any evidence of neonic residues within the resources that bees use. So the systemic translocation is the first area of exposure. In agricultural crops, a big event that can lead to exposure is just planting. So when they have all these seeds in a hopper and they're all, these purple seeds and they're coated and they're getting punched into the ground, that seed treatment becomes airborne. It can leave that seed. And when you're talking about this many seeds, it's a lot of seeds. So soil contamination, contamination of non-target plants, so sunflowers or whatever. So a main route of contamination with ag systemics is during planting and having that many seeds in an area where that dust is released and it lands on soil. And then you also have to think about, every few feet over thousands of acres is a seed with a little wad of systemic insecticide and fungicide on it, right? So that's in the soil. That's where all of our bees are nesting. So what are the implications there as far as just mass contamination of soil? It's there. In the nursery, in a nursery setting, I certainly, I've gotten plants from Home Depot before and they say, this is treated with neonicotinoids, and it's usually a house plant that you don't put outside anyway, or sometimes it is, but,
Right.
I don't know if the systemics are translocated up to the vegetative tissues.
Well, would merit be considered a system? I mean, merit is a systemic. I can't remember how you pronounce it, the chemical, but is that something that would be a neonicotinoid or is that a different?
No, it's not a neonic. Yeah, I'd have to look back and see what that is.
Yeah, and I'm wondering, because we use that a lot, and I would hate to think that I might be hurting bees.
Yeah. Well, I can tell you real quick. I forget what. I forget what chemical class that is.
Yeah, I can't pronounce it.
Okay, so I take that back. Merit is an imidacloprid. So imidacloprid is one of the neonicotinoid compounds.
Yes. Okay.
Yeah, so it is merit is a it's a neonicotinoid. So yes,
wow.
The problem there, is just the presence of that dust in the, in the air, in the soil. Whether it contacts bees from the plant itself, from, the tissues, who knows? I don't know. I mean, I could, it's certainly an important question though, especially if that plant's going to be placed outside or, if it's a house plant, that's one thing, but then again, why would you have a systematic treated house plant that's indoors,
Yeah.
Yeah, it's an issue. And I don't know if anybody's looked at neonic residues in nursery plants, but If not, you know, it's probably not a good thing because it certainly could be a source of stress.
Hmm. Okay.
Have you ever looked at nocturnal insects on Nama hispida?
I have not. I have not, but just this past summer, we've started collecting all the non-bees. And we're finding some, you know, some show somewhat of an affinity for the plant. It's, you know, the plant is so tough that if it has a good bloom year, which there's a lot of variability year to year on when it blooms and um, where it blooms. But no, I haven't looked at any nocturnal insects, but that's a great idea. It would be easy enough to do. But we have started expanding beyond the bees. So now we're asking, okay, what, you know, since this is such a prominent blooming plant and it's a tough plant, and when it blooms, it really blooms. It's like annual broomweed, you know, late season. It has insects all over it. You know, this is another one. It likely does, you know, might have some interesting nocturnal critters on it.
Does anyone else have any more questions? You can unmute yourself and chime in if you wish. Well, thank you so much for helping us out and telling us about your research and pollinators and bees and monarchs. This was absolutely amazing. I loved it.
Oh, thank you. Thank you. I appreciate all the kind words. And yeah, if you know, please feel free to email me or call. If you want to, the land, the little patch of wild land we've been looking at out there, it's gated. They put a new gate up when they built the transfer station. But if you contact me directly and want to come out the next time I'm out there, I'd be happy to show you around. You know, it's not, we call it Walden Plies. It's certainly not Walden Pond, but it's our own little chunk of wilderness. So we love it. And it, you know, when I'm out there, it kind of, I grew up stomping around the wilderness in Arkansas, and it kind of, I can find me little places in this little patch of wild land and kind of hunker down and pretend I'm back there. And you know, it's a neat little place. And we have big plans for it if they would give us the green light. You know, you know, there's a lot of noise from 4th Street. Luckily, there's a big row of elms there on the south side kind of block some of that noise, but we've even thought about, you know, recording sound and slowly restoring the area to, you know, with vegetation over there on that side to kind of reduce that maybe. And, you know, my goal is basically to take as many students there as I can and use it for a good little outdoor lab, which I think, and just simply in that, from that perspective, it's a valuable piece of land. But anyway, if you ever want to go out and take a look at it,
Yes. Let us know if you ever need help with any projects. We are always happy to mobilize and help.
Cool.
Honestly,
Cool.
honestly.
Okay. And maybe when we get these objectives of the NEMA, Hispidum, and Scotosoma prati studies lined out, you know, we might have a component in there where we, you know, meet with master naturalists and, you know, charge everyone to look at a certain area and maybe compare that to bee bowls, or, you know, there's some different things we can do there. And, you know, it's been one of my, it's been a pleasant surprise because I've never worked with master naturalists, you know, to learn about y'all's group and really, you know, I think I met some of you a few years ago, just to have your enthusiasm and passion for nature and you know, and learning about it and, you know, protecting it. And that's pretty cool to know. So thank you all for your interest.
So much for your time.
Yes, thank you.
Thank you.
And just a reminder to all the master naturalists to enter your hours in the VMS and to visit the American Windmill Museum on Saturday. Thanks, everyone. Thank you, Dr. Longing,
Thank
again.
you, Lena, so much. I appreciate it.
Bye.
Bye.
Bye, everyone.